but let's talk about the spatial biology revolution to get started. I think I probably don't have to describe perhaps in this room how fast spatial biology is growing and how young a field it is. Unbelievably young field. I'll show you the whole history on one slide. But the impact has been tremendous. And I think one reason spatial biology's impact has been so strong and its growth so fast is because the medical applications, the diagnostic applications, the drug discovery applications are so close to spatial biology. It's not like next generation sequencing where they had to invent a whole new batch of diagnostics. In spatial biology, they naturally progress to the clinic. And that's the whole reason why the spatial biology revolution is happening and why I think it's going to be so important for changing healthcare in the future. I've been involved in a number of the spatial atlasing projects, and what I'm going to highlight, and it really is, the atlasing started out as a single-cell RNA-seq style project, and what you find is now all the atlasing is now moved to spatial atlasing. Okay? And this one over here on the right is one I'm personally involved in. It's called the SAHA project. And we just released the first release of that atlas a couple of days ago. I think I have that here. So if you take a look in the bioRxiv, you can see the first release of the SAHA spatial atlas. And the reason why I think this is so important is probably most of you study some disease. You're trying to cure something. And oftentimes what is lacking is an atlas for healthy human tissue. So this is the reason why we created the SAHA. It is an atlas of healthy human organs that you can use for a reference. So if you're studying kidney disease, well then you want to find a normal kidney, you go into the atlas, you can see the different organs of the body that we've currently accomplished there. And this is the very first release, so this is going to be a 3 to 5 year project. So it'll continue to grow, but keep your eyes out for healthy tissue atlasing, where you'll have CosMx data in there, genomics data in there to use as reference. Okay, this is a slide. I've done this slide a number of times and I'm still amazed. This is the entire field of commercial spatial biology. 6 years ago, I was announcing this, the very first integrated spatial biology system. It was built, it was called the Genomic system. And when I gave that talk at the American Association for Cancer Research, we were doing 84 genes in space. 84. And that was 20 times more than had ever been done before. So that was great. It was 84. Within 2 years, we went from 84 genes to 22,000 genes in space. Okay, so that was quite a progression, going from 2019 to 2021. And that was building genomics. And then I was also building an imager at the same time. It was called the CosMx Spatial Molecular Imager. It actually started out, believe it or not, as a sequencer, and we turned it into a spatial sequencer, and that is CosMx. We launched that in 2022 with 1,000 genes in space, and at that time it was about 5 times more than anybody had ever done before. And now, just this last year, we are now imaging at subcellular resolution the entire protein coding transcriptome. So that's 19,000 transcripts, all imaged at subcellular resolution. And the whole thing, it's 6 years old. That's it. That's the whole field of commercial spatial biology is 6 years old, which is nothing. Think about how much has happened in the 6 years. So if you project 6 years into the future, you can just imagine where we're going to be at. And I'll try to give you an idea of some of the places we're going. Okay, when I was working on, how did I start working on spatial biology? I started working on spatial biology in 2015. Okay, Spatial biology wasn't even a word. No one had talked about spatial biology. And I was working with a group at the MD Anderson Cancer Center. And that was when the clinical trials in immuno-oncology was just beginning. And I was working with them, and I realized they were trying to understand who responded to immunotherapies and who didn't. And all they basically, the only tool they had was basically a 4-channel fluorescence microscope. And I told them, you're never going to understand what's happening 4 markers at a time. And that was the origin of when I sat down, and that little notebook pad there was in a hotel room before I gave a talk at MD Anderson, and it was a way to do unlimited multiplex microscopy. That was the idea in 2015, and that turned into both of these platforms going forward. But the heart of it was, how do we solve difficult spatial biology problems? Immunotherapy is a great case of that. And I think we're making real progress on it. So the reason why you have to look at the spatial redistribution pattern, you have to look at the spatial pattern, is because all of immunology is a spatial problem. That's the way your immune system works. It goes into tissues and re-architects the diseased tissue, or it maintains health of the healthy tissues. But it's all about the spatial locations of cell types with each other. And if you don't have that information, you can't understand how they work. So we lived in a world of single-cell RNA-seq, which was a great step forward. It was a great step forward. And if you look at the left side of the equation, that is the end of a single-cell RNA-seq experiment, which is a UMAP of all the individual cells. And it doesn't matter what kind of bioinformatics expert tries to look and decipher that, they will never know this. That's what the tissue's really doing. And this is the way you need to look at it. There's 3 different chemistries since coming when I left, where I was a chief science officer for a company called Life Technologies, and I came to NanoStream at that time. And we basically work off of 3 chemistries, but they're all related. If you take a look at that, the bottom one is our encounter chemistry, and it is a fully diagnostic. It's been through the diagnostics. I mean, I've made diagnostics assays for that are approved by the FDA, and I'll describe that. And it was designed to be effective on hospital-grade FFP tissue. Okay? If you want to change the world, you have to work in FFP tissue, because the entire health of the human planet is in FFP. So from the very beginning, all of the chemistries I worked on were designed for FFP. And the key feature that's common is, look at the business end of these molecules. It's 35 to 50 nucleotides of RNA, a little blue, and then Mother Nature's RNA is in gray. And all we do is hybridization. Nothing else. There's no fancy enzymes, there's no complicated processes, it's just hybridization. And it's hybridization with the shortest region of nucleic acid that will allow you to do the entire transcriptome. That's where 35 to 55 bases is how big it has to be to recognize the entire transcriptome. That's why I made it 35 to 50 nucleotides. It can't be shorter. It can be a lot longer, but when you go longer, your efficiency at working at FFP tissue goes down. So I wanted the shortest region of hybridization that would allow me to do the whole transcriptome. That's where it comes from. And that answer is 35 to 50 nucleotides. That's why it's there. Now you recognize that's our FDA cleared assay on the bottom. You have geomics is the next one up. and then CosMx the next one up above that. It's identical chemistry. Our spatial microscopes run off of a clinical grade assay foundation. We're the only company that does that. Our microscopes are basically clinical grade assay in a microscope. So that's different. There's no other kinds of technologies that have that foundation. So when you want to think about translating it to the clinic, you want to make an impact on disease, I think this is the right chemistry to do it. You don't have to change your chemistry ever. You could discover a new biomarker in CosMx, run a big patient cohort in genomics, and make an in vitro diagnostics and encounter, and you would never have to change the business end of your molecule. It's identical. And that's nice. So you don't find a marker up here and it disappears when you make a diagnostic assay, because it's the same chemistry both times. Okay, some people don't realize this. As I was building those two microscopes, the other half of my lab was doing something totally different. They were building diagnostic assays. So we have over 7,000 publications on nCounter, and we were interacting with medical centers all around the world to try to make diagnostic assays for key cases. I'll show you just one key case. So the one I did in my lab is called the ProSigna. We looked at 2,000 archival breast cancer tissues, and we generated the first ever FDA-approved 50-plex RNA multiplex gene signature assay for predicting whether a woman's going to live or die with their particular breast cancer. Okay, we went on to work with drug companies. We made the diagnostic assay for Merck's drug called Keytruda for their pivotal phase 3 basket trial, which means my assay had to work on every organ of the body, and it had to be reproducible in every organ of the body, and that's what we did. Again, I'm just highlighting this so that when you think about our microscopes, you should be thinking, this is a clinical assay in a microscope. So that's what it is. That's how we developed it. Okay, so let's talk about the different classes of spatial imagers and spatial profilers that exist. You have a full-blown imager, which is like CosMx. You have this unique chemistry in the middle. We call it a profiler. That is genomics. And there is no other class of instrument like it. It's one of its only. So I'll describe how that works, because it is single cell resolved with light, but then you actually probe neighborhoods inside tumors or tissue. And then you have array capture arrays. So it's the only one I didn't work on. And I'll just highlight some of the features of the array, and that's all I'm going to talk about it. It's just, I think it's important to understand, when you measure with an array, what do you measure? And that's, I only got one slide on that today. So the thing about when you're measuring on an array system, Mother Nature didn't make its tissues along an XY coordinate frame. They follow their own coordinate frame that makes sense for your body, not an XY coordinate frame of the lab. Arrays are set up with an XY coordinate system, which is the lab. And the problem with that is that your cells don't line up along a grid. And I got there, that's a typical grid for a Beechem HD experiment. And you ask yourself, on average, and I got a real tissue slice on that thing with the segmentation pattern. And if you actually count up how many cells are in a typical grid on an array-based capture, on that particular case, there is 7 cells in that grid. And what you find that that's true in general. You'll go from roughly 3 cells in a grid up to about 15, but you've always got a mixture. That's just the way it works. With Mother Nature, even when you're small, it doesn't line up on a grid. And then I'll show you what an imager looks like on that grid. An imager looks like that on the grid. Every RNA that I work on is 40 nanometers in spatial resolution, so it's 2,500 times better spatial resolution than array capture. And that's the best case. If you actually use them, that's using one element of an 8 micron square. So if I use one 2 micron bin, which you don't really see much signal out of an array, there's 2,500 of these little boxes in that one grid. So it's important. The spatial resolution is totally different league. The other thing to realize on an array-based method, you don't know what's inside a cell or what's on top of it, because it's a 2-dimensional projection of all the RNA or proteins onto a plane. So I don't know whether the RNA is inside the nucleus, is it inside the mitochondria, is the virus I'm measuring inside the cell or on the cell surface? Those are the things you cannot answer on an array-based approach. Okay, so let's talk about some spatial biology instruments. I'm going to really focus this one on CosMx, and then later on you're going to see me talking about geomics, and then we're going to end up on the cellscape system, and I'll try to describe to you how these things work together. Okay, so I think I sort of introduced you to all these concepts, right? From the very beginning, starting in 2015, I knew you wanted to do, felt that you had to do protein and RNA together because we don't know how tissue works. If somebody tells you they do, they don't. We do not understand how tissue works these days, which is great. That's what the spatial biology revolution is going to do. We're going to rewrite the textbooks on how organs work. You need FFP because the goal of spatial biology is cure disease. That's as simple as goal as that. That's what spatial biology is here to do. And then spatial biology is big data. So you really have to generate an infrastructure. So we spend a lot of time helping to generate an infrastructure. It's called atomics. And I'll describe a little bit about that. Okay, so take something as beautiful as this. That's a single human lymph node over there, about 1.4 million single cells. And you image, you can see the beautiful germinal centers, you image it as subcellular resolution, and that's the way you study a lymph node. Now, I'll have to start off with a couple of comparisons because it's important. This field is still so young. It spends a lot of its effort, believe it or not, just comparing one platform against another one. Okay, that's just the way it works when a field is brand new. So I don't have any problem with it. And this field is easy to evaluate. Don't let somebody think you can't figure out how good one platform is over another, because there's only one number you need to pay attention to, 2 numbers. How many transcripts do you get per cell? Okay, and what is the gene diversity you measure per cell? It's the only 2 numbers that are important. How many transcripts do you have, and what's their diversity? That's all you got to do. So because if you don't have that, you have no information. If I take the transcripts away, there's nothing to measure. And it doesn't matter who you compare the current ones we make with whatever system you want, you're going to see data looks like this. This will be the CosMx data, and every other platform is going to have their little violin plots at the very bottom, because they don't measure many transcripts. Okay, and there's a reason for that. And it goes back to the original chemistry, okay? So I have this little simple saying, hybridization-based method. Hybridization scales, whereas enzymatic fail. At high plex, that's the way it works. Hybridization will scale to whatever plex you want to take it to. But enzymatics in tissue are very complicated, and it just does not scale to high plex. That's why we never did an enzymatic approach. And there's a reason for that. So if you do something like a rolling circle amplification in tissue, what happens is there's a polymerase that's got to spin around and make a whole bunch of copies of something. And as it spins around, If it's in the nucleus of the cell that's very crowded for proteins all around, it spins at a certain rate. It goes into regions where there's not so much dense, it speeds up. So everywhere you look at in a tissue, those little rolling circles are running at different rates. Why does that matter? It matters because the thing you're measuring then varies upon where you happen to be in the tissue. So if you're measuring a single RNA spot, the size of it matters. It's going to grow for those areas where the rolling circle is spinning fast, and it's going to be smaller in the regions where it's not. And the fluorescence intensities are going to be going up and down. And they go up and down in the best case scenario, roughly 3x is what you get with fixed cells. Tissue is an order of magnitude worse. These intensities vary widely. Whereas with your hybridization, you don't have any of that. Every spot you measure on the CosMx is same exact size, it's 20 nanometers, and the same exact fluorescence intensity, 30 fluorescent dyes, because I make them in the factory. And everybody in the world gets the same thing. Okay. So when you do an experiment with a hybridization-based approach, which is the upper, it doesn't matter what plex, you do the same thing. You hybridize all your probes, either 1,000, 6,000, or 19,000 all at one time. You put your segmentation markers on, and you go into the instrument and run. If you're doing something like a rolling circle amplification, that's not what you get. Yeah, all those little stars are, when you go to high-plex rolling circle, you got 3 new phases you have to do: an extra primer hybridization, an extra RNase, an extra polysine. Then you hive all over again, then you ligate, then you rolling circle amplify, put your segmentation markers on, and then you're ready to read out. So that whole 2.5 days first step, it's worth asking yourself what's happening during that first 2.5 days. Those first 2.5 days, every individual that's running that experiment is making the imaging reagent on the tissue one slide at a time. That's what that is. You're making the imaging reagent one slide at a time. Whatever technician happens to be running that sample, they're making the detection reagent on the slide. It's not very good for a clinical style assay if you want reproducibility around the world and you want to get the same number no matter where you're at. Yeah, that's not going to give you the same number. Okay. A lot of people said it was going to be impossible to image the whole transcriptome. In fact, almost the entire world told me that we weren't going to be able to image the whole transcriptome. So I had to prove that we do, and it's not that hard when you got the chemistry right. So look at the one on the left-hand side is the key one. So I'll take a slide and measure it at 6,000 plex. Okay, that's the x-axis of that graph. I take that same slide and I put 19,000 probes on it, and I measure it over again. And then I just plot of those 6,000 overlapping genes. Was the numbers the same, or did they change between 19,000 and 6,000? If you have a perfect assay that scales ideally between 6,000 and 19,000, you get a straight line, slope 1.0. What do we get? Pretty much a straight line, slope 1.04. Okay? So what that unequivocally answers is that we scaled the assay from 6,000 plex to 19,000 plex with absolutely no non-idealities. It's totally an ideal assay. 6,000 plex to 19,000 plex without a single point off the slope 1 line. So no one can argue, well, you can't measure 19,000 at a time. You absolutely can, and you can do it perfectly. Okay? So some people will say, well, then you can't locate them. You can't locate 19,000 things at a time. So that's what the other graph is, this one over here. So we measure it. We measure how accurately do I measure every single dot that I measure, and I typically measure about 6 billion at a time. And that's the spatial distribution of them. 38 nanometers. That's the mean of that spatial distribution. And if you look at our original peer-reviewed paper in Nature Biotechnology, 2022, and we plot that out for 1,000 plex, it's identical. You can look at the original Nature Biotech paper and 1,000 plex, look at this one, it's the same thing. So I went to 19,000, no change in the assay, and I maintained the spatial localization. Nothing changed. That's important. Okay. So you don't have to take my word on the sensitivity comparisons. There's a lot of them out there. If you want to see one and the talk and everything, just take a picture of that little QR code. But what's important is if you're going to do a comparison, you got to do it correctly. Okay. There's a lot of noise out there, but just look at a well-controlled study. They got 16 patient blocks here. This is all associated with inflammatory bowel disease, so this is in the colon. 16 different blocks, 11 different patients. And they come out, they go to the same person that's sectioning for CosMx is the same one that does it for Beechem. 5 micron FFP section, boom. At the same exact time, one goes into a Beechem, the other one goes into a CosMx, and you just look at the sensitivity numbers, what you get when you do it in a well-controlled manner. So I'll show you what it looks like. There's 3 different assay plex regions, okay? One of them is 1,000 plex. That's the lowest plex assay I made. And there's another one at 6,000 comparison, and then there's another one at whole transcriptome comparison on the next page. So when you're way down there at CosMx 1,000 plex, and you're looking at it on a custom Beechem panel, we're 5 to 6 times more transcripts. The violin plot's 5 to 6 times higher. The overlapping genes are similar, so 1.2 to 1.8, I think they're pretty much similar. Now you can say, well, that's just because you got more plex, Beechem. Isn't that the case? Well, let's look at the next one. That's 6,000 plex versus 5,000 plex. They should be within 20% of each other, right? They're not within 20% of each other. We're still 4 to 5 times more transcripts than they get at 5K that we see at 6K. And then when we measure the overlapping genes now, there's 2,550 of them. We're now 2.5 to 3 times more sensitive on all the overlapping genes. So we're really getting to a place where the only comparator, there's nobody else out there measuring these kind of transcripts. The only comparator is single-cell RNA-seq, and that's sort of what you see over here. And it's sort of an interesting experiment, I think. Over on the right-hand side is almost 500,000 cells on a chromium single-cell RNA-seq experiment, and that's the gene diversity that they get after almost 2 or 3 years worth of work. And then the very first time they put CosMx whole transcriptome on their colorectal of their inflammatory bowel disease sample, they plotted out what they got after 1 run. After 1 run, they had more gene transcripts and more gene diversity than almost more than 500,000 single-cell chromium experiments. We actually saw more genes in 1 run than they did in about 2 to 3 years of single-cell chromium experiments. They were pretty happy about that. Okay, then this is what it looks like at whole transcriptome. Nobody's with us at whole transcriptome, so I can't do any direct comparisons. You'll be averaging about 1,550 transcripts per cell, and you'll have a gene diversity of roughly 900. That's averaged over 6 solid tumors in the body. There's a recent paper that came out with over 1,000 samples from Beechem on it, and these graphs are to scale. So 5,000 plex Beechem is going to give you roughly 50 to about 350 transcripts per cell. That's over 1,000. And we're up there. So there really isn't a ton of comparison. The other thing that's important to note is, what's this number here, this thing called DV200? DV200 is a sort of a record of the quality of the RNA coming out of a hospital FFP process. So when we designed CosMx, we made this thing a flat line. So it doesn't really care what the DV200 is. It's going to give you roughly the same numbers And that's in the original He et al. Paper if you want to take a look at that. And then here is what a rolling circle looks like. Rolling circle is incredibly sensitive to where you got your FFP tissue, what hospital did it, what they were doing that particular day. And we're flat. So having a slope like that is not what you want. You want your slope to be flat for TB200. Okay. And then there's just one more on here. We continue to increase the sensitivity assay. If you upgrade to our new CosMx 2.0 and Atomics 2.0, every assay you're going to do is got 1.5 to 2 times more sensitive. Without doing anything, every time you run again, it's going to be 1.5 times to 2 times more sensitive, and it will occur in about half the time. So that's kind of a nice add. Okay, let me say one more word, and then we'll get on to the biology. This is still technical kinds of things, but they're important, I think, to understand the technical things. This has to do with when you segment a cell, you want to make sure to get all those transcripts into the right cell. And we've been pointing this out I've been talking about this for about 6 years. This is the dominant error term in imaging, high-plex imaging. It's this term. Forget about all the other ones. This is the one that dominates. That's why we work so hard on it. We believe in data-driven segmentation. We have been from the very beginning. We're on our 5th generation segmenter, and I think it's best in the world from the commercial perspective anyway. You know, when we're in a brain and we're measuring an astrocyte, the segmentation looks like an astrocyte. It doesn't look like a round ball. Those astrocytes are round. And look at the resolution we have on organs. I'll show you some more. We've now trained. It's a cell pose engine, but it's all our own training on top of it. I've literally measured 2 billion single cells on CosMx so far. And we train on that. I wanted to show you one here because it's a brand new segmentation. I think it's the only one in the world that will handle this particular case at this current time. When it comes to cardiac muscle and skeletal muscle, these are very difficult things to segment. The cells are totally unusual. They are multi-nucleate. Some of them can be huge. They can be millimeters long to single cells. They're multi-nucleate, and they have very complex patterns. And with Atomics 2, we took something best in the world with something looked like that. That was us, and it was bad. And now we're over here. You can see, I don't know whether you can see from the back of the room, these are beautiful plasma membranes. You see these little areas in here? Those are important cells that are interspersed between those large, monstrous cardiac myocytes. So you have to be able to resolve little tiny things, huge things, and multi-nucleate things all at the same time. And we did that. And it's available for you now as a pull-down menu. I think it's the only place in the world you can actually do that with cardiomyocytes and skeletal muscle. These are really beautiful. There's a skeletal muscle that's actually had some trauma. It's got huge immune infiltrates there. And look at how, maybe from the back of the room, we're segmenting all through the immune infiltrate, all through all the major fibers. So I'm pretty excited about that one. It's brand new. I'm going to show one more. Okay. These are these really unusual. See those little blue dots? Those are the multi-nuclei in skeletal muscle fiber. Look at what Mother Nature does. It pushes them right up against the plasma membrane like that. Yeah, we find them, we segment on them. Very unusual type, but super interesting from a spatial biology perspective. This is one that just came out. And I get bone marrow is an incredibly interesting organ of the body. I view it as an organ, we have one group now that have been looking at bone marrow samples long enough. They have generated over, you can see those numbers there, 448,000 nuclei, 320,000 cell membranes, all in the bone marrow. They took our CosMx cell pose engine, fine-tuned it for bone marrow, so you can inherit that. Then all of a sudden, they're doing these amazing studies of bone marrow. These are time progressions. This is in leukemia, and we are resolving all these kinds of segmentation processes. And you get these really odd cells in the middle there. I think they're called megakaryocytes. So you gotta recognize them, you gotta cell segment them. So if you do some bone marrow stuff, there's some really neat segmenter. It's all CosMx based, not done by us, so it's done by the National Cancer Institute. We work together with them to help do this. Okay. Now let's talk about biological discovery with this thing now. This is really what it's all about. This is inflammatory bowel disease. This is actually clinical trial work done at Northwestern. And the Northwestern MD physician, you know, he's being funded by drug companies to do these clinical trials. So he had to drag along basically Beechem and CosMx for a while, and then now he doesn't have to do that anymore. The pharmaceutical company said, yes, you don't have to do that anymore. And one of the reasons is this. They made a key discovery in inflammatory bowel disease, this molecule called seleno-P, and it showed up in the CosMx data. It was invisible from the Beechem data. They didn't see the discovery at all. And then that was the same group that had 500,000 cells of chromium, and they did all the receptor ligand interactions off that single cell dissociated, and they had all these that sit down there. And they were actually, the drug companies were looking at them, trying to figure out how to get drugs for these different ligand receptors. When we did it at the whole transcriptome level, all those little red dots there, those are all false positives. None of those were real ligand receptor interactions they got from the single cell, because dissociated single cell doesn't know what a cell over here is. The cell could be 1 centimeter away, and the analysis in single cell was saying, this guy's talking to that way. You don't talk over a centimeter of space, but when you do the analysis, it never knows. So they all look equally plausible, but in tissue, that's not the way it works. So now the drug companies are focusing on the LRs that we found. Those are the real ones that are happening in that tissue. So whole transcriptome allows you to do that, which is important. It also allows you to discover things that no one expects. And this happens to me almost every day I measure on CosMx WTX. And this is an example. I'm in inflammatory bowel disease. And what do I find is one of the key pathways that are altered in early stage IBD is olfactory receptors. It's like, when I found that result, I actually called up a good friend of mine who was one of the people who studied olfactory receptors for over 30 years, a guy named Peter Mumlars at Max Planck in Frankfurt. And I said, we just found dysregulated olfactory receptors in the colon for IBD. And I said, I don't know what happened. And he said, oh, Beechem, they're in there. A good friend of mine discovered olfactory receptors are in there. And when I went and looked what they are doing in there, they are measuring short chain and long chain fatty acids associated with bacterial metabolites that are happening in the colon. So then it's like a light goes on. Okay, that's why that pathway is up. But the key thing is, if I would have just examined inflammatory bowel disease with the knowledge as it's sat, and make a little custom panel with 500 plex and say, I think I'm covering everything that happens that's important in IBD, I'd be totally wrong. There's not a single olfactory receptor that's ever been done for a custom assay in the colon in the literature. I don't think it's ever been done by anybody before. But when you're at the whole transcriptome level, I don't make any assumptions. It's totally unbiased discovery. And every time I measure, I see stuff like this. Almost every day I measure. Remember, the field's 6 years old. No one's had time to discover all this stuff yet. But it's an opportunity for you to do that. Okay. And it's another excisional biopsy on the same IBD sample. These different things all end up working through a very interesting pathway that shows up in cancer all the time. It turns out that same pathway is active here in IBD. It's called ferroptosis. If you just Google that, you'll see it all over the cancer literature now. And here it is in IBD. Okay. This is the part of the talk I like the most. when you measure at the whole transcriptome level and you start thinking about the data analysis, it totally changes how you think about analysis. Okay? That's an excisional breast cancer sample we're looking at here. And when you work with a low-plex panel, like say 500-plex panel, what you're actually doing is cell typing. That's what you got. At 500-plex, all you can do is cell typing. It's good, nothing wrong with cell typing, but you got to ask yourself, why am I spending so much attention to the cell types? You're doing that because you are inferring the biology from the cell types. That's what you're actually doing. Oh, it's an M2 macrophage, that means it's tumor suppressive. Well, yeah, maybe. But that's why you're inferring the biology from the cell types, because that's all you got. When you're at whole transcriptome, it's unbiased discovery. So you just see all the biological information at one time, even before you cell type. And let me show you an example of that. Let's just take a look at this thing, and I'll talk to you about how you analyze it at the whole transcriptome level. It's totally different than what you do at 500-plex. And I think so much easier to understand at high-plex than at low-plex. So here it goes. Every time you measure a patient sample, there's that same breast cancer sample, I'm measuring about 2,700 pathways at once. Okay? So then you can just say to yourself, let's just plot the pathways in space. I think everybody that's been doing this for a while in this business understands things like MAP kinase pathways, TNF alpha, JAK-STAT. You just measure it and put it on the tissue. That's what MAP kinase is doing. That's JAK-STAT, that's TNF alpha, that's the hypoxic HIF genes coming up. I'm not even cell typed this thing, but I have all that biological information immediately available to me. Okay, let's take it one step farther. There's that same excisional biopsy, there's a protein in it on the side so we can really go into it a little bit. And what you can do is take a look at all the biological pathways in space and watch how they change in space and group them into domains of activity. Okay? You take a look at, and this is what the domains look like when you do that. Let's take a look at that TAN domain. What is that? I look at all the pathways that are linked in that spatial pattern, and I can just read off what that is. That is the invasive front of the tumor. Okay? That is proliferation and invasion. I didn't even cell type this thing, and I know where the invasive front is. To me, that's one of the most important things. You want to know what's happening. Where's the invasive front? Where's the war happening? I did it. One fell swoop. No fancy bioinformatics. Just what's the pathways active in every cell in that system. That's what it looks like. Okay, what happens right behind it? So there's what's right behind it. I said, what are the pathways right behind that invasive front? And that's what this thing is. I call it looks like a train wreck. Okay? Nutrients are gone. Stress responses are all up. This is like a disaster right behind it. So you got the invasive front comes through, and right behind it is basically no nutrients left. It's stress response all over the place. And that's not a bioinformatically inferred thing. That's a directly measured thing. Okay? So let's look at what's the cell type pathway changes look like. Yeah, that's pretty neat. So let's look at where the action is, right in there. And let's take a look at an important cell type, activated CD8 T cells. You know? That one's either going to live or die, depending on what our T cells are doing here. So let's find out what they're doing there. Those little black dots, that's the external stromal domain up at the top. And then what you can do is monitor the pathways that happen to the T cells as they go into the bed. What happens as they do that? I just read the pathways off, it's easy. As they go into the bed, the metabolic activity of them goes skyrocketing. So all of a sudden, it's like everybody turned on the gas on the metabolism, and that's what's going on. Boom. And then what else is happening? As the metabolic pathways go up, the tumor is turning off their survival mechanism. So it's like being in your car in neutral with the accelerator on the floor and somebody's pumping the gas out of your car at the same time. That's what's happening in that invasive front area. And then one thing nice is that what we discovered, one of the key things that turned off the T cell survival is a molecule called GMAP2. I looked in the history of everything I could find. By anybody ever look at GMAP2 in cancer before? A spatially resolved manner, the answer is no. No one ever looked at GMAP2. GMAP2 belongs to a large family. And when you're making a little panel and you see a family of stuff, you got to make a selection. Which GMAP do I take? It turned out that this one must not be in PubMed, and nobody did a literature search. You never find GMAP2, so it never goes into somebody's panel. Well, for this woman, GMAP2 is the key thing that she's got to worry about. The whole transcriptome, I never have to make a decision. What family member of a gene class do I do? I do them all, no matter what. That's sort of another example of why it's important not to look with little blinders on. Okay? So that's sort of an example of what you see at WTX. I thought I'd give you a couple of other discovery examples just to impress upon you that we can't make little panels yet. We don't know enough about the tissue to do that. And this is another example of where we found something totally unexpected the very first time we measured. The very first time I measured pancreatic cancer at high plex, this was at 6,000 plex, with a very talented physician scientist at Harvard named Will Wong. We ended up with a whole batch of spatial correlations. And we're down here, it's one of the strongest spatial correlations we found in that sample. And you know what it was? Something that nobody ever looks at in their old cancer panels. It was the nerve cells and the stroma of PDAC talking to the tumor cells, talking to the immune cells in an active, participatory, regulatory way. It was the nerve system right in the middle of PDAC signaling like crazy. No one was putting nerve cells into their little panels for custom cancer, but I didn't want to make that assumption, and they were there. One of the strongest things happening. And I remember giving this talk at the American Association of Cancer Research. At the end of me showing this, I had like 10 medical doctors up at the front of the stage. And what did they want to tell me? Oh, I knew there was something like that, PDAC. I said, how did you know that? Well, we do a thing with our pancreatic cancers. I palpate their pancreas. That's what I do with my hands. And if a patient feels pain upon palpitation, their prognosis goes way down. That was their assay, their hand. They never knew why. Yeah, that's why. That's why. And now we understand that. Once we understand that, we can start developing a drug that's associated with that, and that's where that whole group is going. They recapitulated this response outside of the patient. They got co-cultured primary tumors and nerve cells interacting, and they actually see some of the same exact signaling patterns. They isolated a small molecule that we predicted would be there, and they found it, and now they're talking about making a drug to it. All within about a year and a half. So that's kind of exciting. That's what we built these microscopes to do that. This is another one I like. This is another discovery. Different kind of discovery. This is a spatially resolved discovery. This is an esophageal brain cancer, esophageal metastasis to the brain. This is work we do with MD Anderson. MD Anderson were one of the leading institutions that made us aware that the microbiome plays a role in cancer and in cancer immuno-based therapies. So it's important for them to understand it at a fundamental level. So that's what we did with them. That's a CosMx three-dimensional image of that cell right there in the middle. And you see all those little red dots in there? Those red dots are 49 intracellular bacteria in that tumor cell. Okay? Now you can go to other areas of the brain and you can see all these bacteria, but they come from the processing of the FFP. And you gotta disregard that. But that's intracellular. That's inside the cell. On an array, I would never know what's inside or outside, right? Because I just plastered it down into one two-dimensional array. I'd never know what is the bacteria inside versus outside. It's only the bacteria inside that matter. And that's unequivocal proof of that, and we found that. We looked at a bunch of tumors. Over 95% of the bacteria are in the tumor cells. People didn't know that. They thought maybe they're in the immune cells too. They're in the tumor cells. And we're writing this up now. Okay, talked a lot about RNA, but I love proteins as much as I love the RNA. This is a great example of a clinical result that comes from subcellular localization. That's a corneal biopsy from a breast cancer patient there. And that patient is in a clinical trial program that will be combining immunotherapy with tyrosine kinase inhibition. Okay, so those are the two things. And we are the arm that determines whether that patient has activated phosphorylated ERK 1 and 2, which that drug actually operates on. So here's what we saw when that patient came to us and when we measured it. Phospho-ERK, when it's activated, is nuclear localized. Okay? And when it's non-phosphorylated, it's out in the cytoplasm. So if you look down here, the little red ones, these are in nuclei. And then the stuff out here, that's non-activated ERK. And I see them both, and I can quantitate them both. This is CosMx protein, as they know. So I told them, oh, this woman has a lot of phosphoactivated ERK 1 and 2. She was lucky enough to go onto the arm that was the tyrosine kinase inhibition. I have a longitudinal, I have another corneal biopsy a couple of weeks after she took that drug, and all the phospho-ERK is gone. So she went onto a tyrosine kinase inhibitor and it did its job. So that was good for the MDs. They said, okay, I don't have to worry about that. I have no idea how it will synergize in a clinical trial. I'm blinded to that, so I won't know that until I report out. But unequivocally, that side of the arm was doing what it was supposed to do. And this is an example where you could, I think, only absolutely unequivocally do it with protein. And it has to be subcellularly resolved protein. That's what we got. Okay, you don't have to believe me. I'm not going to go through all the details of how we make these molecules, but it's not very complicated. It's all written in this paper. And you can edit your whole transcriptome with an oligo that's about 110 bases long. And you can read all about how we did it in there if you like. I'm going to focus on some other aspects of it. When we did this assay, I wanted to be able to be as good as single-cell RNA-seq, but I wanted to maintain the XYZ coordinates of everything. Okay, so that's a big task. And when you do this, you do serial section, single-cell dissociative RNA-seq, this is what you'll see. The CosMx distribution is the blue. The single-cell RNA-seq is in the red. Ours are completely symmetrical. You don't have any asymmetries. You don't get any doublets or triplets, and that's what you see in the dissociative stuff. But obviously we're measuring hundreds of times, thousands of times more cells in one run than you will get out of a dissociative experiment. So we did the most important thing was match the gene counts and the transcript diversity. Doesn't matter what cell type you're in, we match it all the way along. So that was very good. That was really a hope of mine. We were there. And I was giving this talk down in, I think it was, I don't even remember what city I was in, but somebody came in out of the audience. They are now 600 patients into a non-small cell lung clinical trial, and and they have CosMx whole transcriptome on 600 patients. That was great. The first time I got to see the data, this is what it looked like. It's done in 2 different hospitals, one in Queensland in Australia, and one in Yale in the United States. Here's their signals. You can see the patient distribution here. Here's their positive signals out of the whole transcriptome. There's their negative probes here, and these are things called false codes. These were performed halfway around the world from each other. They put the data together, and it absolutely harmonized along a perfect straight line. So that's what you did along that side. It didn't matter what hospital I was, it was halfway around the world, and the numbers they got in Australia matched the numbers they got in New Haven, Connecticut. And that's how clinical assays are supposed to work. And that's why this one, now you understand why this one did work that way. Because it's no different. It's the same as our clinical assay hybridization things we built years ago. Just a microscope with the same properties. The other thing you'll find when you do this kind of work is that you can see rare cells that are invisible almost any other way. This is an example of a pancreas here. We have these rare cells in an islet called epsilon. Every time I measure a pancreas section, I'll see about 130 of those cells. They're at 0.008%, but they're important. And if you do that experiment on a chromium, and we did it, it's just invisible. You'll never see them. I don't know whether they don't make it. There's a whole bunch of cells that don't make it to a dissociative experiment. Maybe it's one, or maybe it's just so rare I have to do 10 million cells to see something. But here, they come out real easy. And the other thing you notice is that if you look at the cell distribution from a spatial experiment compared to single cell RNA-seq, I think most people know this, but the cell distribution you get from single cell RNA-seq is all wrong. It does not represent the cells in the tissue because not everything comes out. This is a colorectal sample I'm looking at here, and single cell RNA-seq says it's about 3% smooth muscle. I'm looking at that section, that FFP section, I can see with my eyes it's about 20% muscle there. They don't see anything. And CosMx, of course, is going to see every one of those. Yeah, because you don't have to dissociate them. They're there and they're being counted. So the same thing happens with a whole bunch of other cell types. The eosinophils don't disappear in CosMx. The neutrophils don't disappear. So it's important to keep in mind when you're making these comparisons, you try to figure out which one's right. Yeah, the cell population distributions in single cell are not what is in that tissue. I think most people know that, but I'm just sort of highlighting it just to emphasize that fact. Okay, let's do something totally different. Okay, let's not do a tissue experiment. Maybe I'm doing something else with a whole transcriptome image. Maybe I'm doing mixed co-cultures. Maybe I'm doing organoids. Maybe I'm doing 3D cell cultures. Maybe I'm doing CRISPR-Cas9 experiments. What's it look like when you do it on an imager? These things are amazing, so let me show you. This is what it looks like. I'm imaging now the CRISPR-Cas9 guides that are in these individual cells that are doing surgery on the molecular motors inside those particular cells. That's what CRISPR-Cas9 does. So now I can monitor the effects of CRISPR-Cas9 in the whole transcriptome. So I'll see every biological effect every time you touch a promoter. Okay, what's it look like? I've actually been doing this for about 2 years with a drug company called Merck in my lab. And they're so anxious to publish it, they now let me talk about it so I can get to show you what it looks like to do high-level drug discovery using a CosMx imager and CRISPR-Cas9 on organoids. This is how we make them. This is for the people in oncology, you'll understand that, and for people not in oncology, you'll have to bear with me. Okay. There's a particular cell type in tumors called cancer-associated fibroblasts. These are called CAFs. And CAFs have a million personalities. They do all these important things to the tumor cells in the stromal regions of patients' solid tumors. And nobody understands them well. Okay, so what are we doing? We're taking those calves and we are doing high throughput CRISPR-Cas9, knocking down all sorts of interesting linear receptor pathways and all this stuff. And I mix them with the primary tumors and measure the behavior of that mixture at high throughput. This is what it looks like. Every one of those things is organoids. I can put about 1,000 on a single microscope slide if I want. I'm still working on this technology. Measure all the guides, all of our stats work in a CRISPR-Cas organoid like they do in tissue, so that's what should happen. And then these are what these organoids look like. Yeah, I think they're beautiful. There's so much information in there. You can see the tumor cells around that, the side, those are the light green things you see there. And then the Cas are associated fibroblasts and some of the guide RNAs on imaging, and they're all got different kinds of behaviors to them, and then I'm measuring that whole thing at high throughput. And I'm doing it multi-omically. So I am engineering them, measuring the whole transcriptome, and then I got about 75 proteins all on the same organelle, all in parallel. You get these beautiful patterns. Look at fibronectin snake its way through that particular organoid, EGF receptor, yeah, all these key protein things. We're seeing at high throughput. And you can see we have an opportunity to understand CAPS at a level it's just impossible to get away, I think. And this gives you an idea how the information comes out. I got my CRISPR identities over there, and then you can see these are all the individual conversations that we're having, that we're knocking down. ITGA3 talking to ITGA1, COL1A2 talking to STC1, so on and so forth. I am just tearing the CAPS apart, and I'm understanding how every little interaction happens with the primary tumor cells. And yeah, I think it takes our knowledge to a whole new level. Okay, I'm not done. Okay. I got one more trick I want to show you. And that has to do with the multiomic aspect of CosMx. I haven't spent a lot of time talking about protein, but I think you'll appreciate it after this little section. It works with all of our panels. And let's just take a look at what it looks like when you do this. This will all be implemented actually mid-year. So if you have got a CosMx with the latest software, it's actually ready to do multi-omic work. We'll help you implement that. Obviously, I've been running Westway for about 2 years now. It takes a while for it to get all the way out into the public. Let's take a look at this. This is non-small cell lung cancer I'm looking at. There's a whole transcriptome image on the left. There's the morphology on the right. There's the morphology image on the left. For reference, the tumor nest, PanCK, is in blue. So you see a little tumor nest up in the upper left, and you see a little tumor nest down in the lower right. And everything in the middle, that's the infiltrate. You're going to live or die depending on who wins that battle. That's the immune system trying to kill the tumor cells right there in the middle. And that's what I'm trying to understand. That's what we need to understand. Okay, so doesn't the whole transcriptome give me everything? Well, no, it doesn't give you everything. Take a look over here. See that tumor nest up there? It looks like there's a gap between the tumor nest and the infiltrate. Whenever you see a gap in an image, you got to ask yourself, Mother Nature doesn't like gaps in tissue. Maybe I just don't see something that's there. So we went looking. I went looking through the high-plex protein that I have, same slide, a single cell line, high-plex protein on that same image. Let's see what we found. It's one of the first things we found. That blank area was not a blank area. That blank area is full of extracellular fibronectin. Okay? So that little tumor nest that's in the upper left-hand corner has built an entire network of protein guarding it from the immune infiltrate. I didn't know that just looking at the RNA. Okay, I guess I would have missed it. How about this other one down here in the lower right? What's happening down there? We went down there. We found another barrier around that one. In this case, it's smooth muscle actin that built a barrier around that tumor nest. Okay, that's some useful information. What else do I got inside the high-plex protein? Okay, you can ask yourself, couldn't I have found it in the RNA? I don't know if you're in the back of the room, but you'll see this. Those are the smooth muscle actin RNA transcripts in little green dots. You see that? If I'm trying to convince you that's a barrier with those little green dots, would you believe me? I wouldn't believe me. Okay? But a single RNA from a smooth muscle actin can make like 1,000 smooth muscle actin proteins. And that's how you get that pattern. But you have to see it at the protein level. You would never really know from the RNA level. Okay, what else can I look at? Okay, this is one I like too. B7H3 is one of the key checkpoints. Okay, and it responds to immuno-based approaches. And look at where the B7H3 shows up. It is right on the boundary between the tumor nest and the barrier proteins. And I remember I was giving this talk the other day, and somebody came up by the audience, and the person said, I always suspected that. I just didn't know how to prove it. I said, well, now you can just see it with your eyes. You don't have to do any fancy analysis. Where is it? Yeah, that's where it was. Okay, let's look at one of the interleukins, IL-18. So I like this one just because this is where the war is happening. Cells are living and dying and giving off stuff like this all the time. Interleukins, cytokines, chemokines, all sorts of stuff. So you got a good battle, active battle happening right there. Nice to know. Here's a protein called STING that was in our high-plex. This is in the existing universal 64-plex base, so this is commercially available to you now. That's the STING. And look at how it lights up these individual cells. It's always amazing to me. You can see something like this just stand out at the protein level right in the middle of a infiltrate like that. So I guess I can talk for about 2 days on this story. But the point, I guess, for you is just I'm trying to convince you that I started out with that RNA image over there. And now I got this same slide multiomic. All that additional rich information is really important. I sort of view it, it's like going to a sporting event. Imagine you go to a sporting event and you can't see the playing field at all. You just see all these people running around. They're playing soccer or cricket or football, who knows what. Until you understand the field that they're playing on, you don't really understand the game. And that's what the protein gives you. The protein gives you the architect of the field in which this life and death war is happening at. And once you sort of see this once, it's like I can never look. It's terrible because it's hard to go back. It's hard to not see the multiomic side of the equation. Okay. You can also do things with same slide proteomics that are even simpler than this fancy barrier. And then I like it helps resolve your cell type ambiguities. People just, I came from the RNA side, I just thought my cell types were perfect. Your RNA cell typing is not perfect. So if you look at the UMAP for protein-based cell typing, RNA-based cell typing, and then you do the integral, where you bring both sets of information together, what I find is that about 70% of my immune cells that I'm measuring all of a sudden have better tuning. I can go farther down into what subtypes of that immune cells are. I can find errors in the RNA cell typing this way. So it's valuable from a simple cell typing also. I'll give you one example. I can give about 1,000, but I like this one example. Look on the left-hand side here, right in the middle, my little RNA in situ types telling me that cell right there is a macrophage. But then I look at the same slide, aligned protein image, that cell's positive for CD3 plus and CD8. That thing's not a macrophage, that's a T cell. It just happened to be a T cell that had a transcriptome that was more aligned with a macrophage than it does a T cell. That's just the way it works sometimes. But I can go in there and correct that one. Sometimes it just confirms there's a macrophage right above it. It stains perfectly, but CD68, so that guy stayed a macrophage. I call it a validated macrophage. But you're building, 5 million cells at a time this way. Yeah, you got a lot of little corrections that happen, but very useful. Okay, this is the one I like sort of the most, I guess. Back in 2015, when I was dreaming about microscopes for immuno-oncology, this is the thing I really dreamt about. I wanted to get the whole transcriptome. I wanted to have high-plex protein on the same thing, and I wanted to have an aligned H&E slide all together on one piece of information. And that's what it looks like when you do that. That's a single 5-micron section from a patient. And I could write a book about that patient with the information I got. I almost feel like I know these patients after I look at these things. But, yeah, now we're getting somewhere. I think now we can make a better drug with this information? I think absolutely yes. Can we make a better diagnostics with this information? Absolutely. Maybe next time I come back, if you want to mark it. Okay. I think it's the last topic here, and then I'll give you guys all a break. This has to do with the infrastructure we built. It's called Atomics. And I knew from the beginning how big these data sets were going to be. They are not, if you think you can just homebrew an infrastructure that's going to work for you, you just, after 10 years of doing this, it's like, yeah. I used to have people come into my office about every 3 days asking for me to buy another server or can they get something else online to look at my little data sets. That really gets old. So we built it all in a cloud. A cloud is unending storage. It's unending compute. Anytime you want it, you just get it. But the goal of getting it onto the cloud was not just that. It had another goal. And we're just starting to get to the place where we got the cloud infrastructure done well enough to help enable it. I had one simple goal. I wanted the biological person that paid for the experiment, probably a principal investigator that's responsible for curing that disease, making the next breakthrough. I wanted them to be able to look at the data themselves and analyze 80 or 90% of it without having to reach for a biostatistician or a bioinformaticist. And why? Why do I care? I think what you find is that the average spatial informatics person doesn't know a kidney from a lung, from a pancreas. They don't know how it's organized. They don't know what a glomerulus is, or an islet is, or a tertiary lymphoid structure. That's not what they were trained in. And they're supposed to give you the answer to how you cure a disease. And I love them, they're great, but that's not what they're trained to do. So Atomics was built so that you as a PI can figure out the disease you're working on yourself, with your own brain, and that's what we try to implement. And that's why it's up there. There's a bunch of things we're adding to it. I'm just going to highlight one term. The term that causes people a lot of trouble in high-plex analysis is cell typing. I'm going to highlight one thing. But anyway, so these tools are all available to you. They're not black box tools. Every tool we put in Atomics we publish on. You'll see the code, so there's nothing hidden there. We do all sorts of pathway analysis and all these types of things. And I want to highlight one. We just did this one and released it in June 20th, so it's only 1 week out to the outside world. But I think it's important. If you're at high plex and you want to do cell phenotyping, you can't just use your little marker gene kind of thing anymore. It doesn't work. At 19,000, and you're using a tool that was built for 500 flags. It don't make that mistake. So this tells you how not to make that mistake. And I'll just show you one little example. If you're taking this, typically people look at marker genes along different cell classes. And then that little blend at the bottom is the T cell marker, sort of like you get when you look up the textbook marker genes thing. Yeah, that's not fine tuning your phenotyping. So if you use this kind of hierarchical cell typing thing that we just published on, and you can look at it and you can download the thing, you actually turn that little box into all these things: CD8 exhausted, cytotoxic, naive, TH2s, T-regs, T17s, delta 1s. You can decompose your data all the way down to a very fine level. Don't use the little gene marker tool. It's never going to get you there. You have to, if you read this, you understand how you make use of the whole transcriptome to do something like this. And that's what's written up. And you get the right answer. What you're looking at the bottom is an area of a tumor that's got a tertiary lymphoid structure. I do all the fine mapping and then map exactly where things are supposed to go. And what you find is that our T helper cells and the B cells all track with each other, and that's the way they work. Okay, your CD8's got a whole different program, they do a different thing. So when we do this, it generates cell types in a tertiary lymphostructure, so it's following the biology that people know about TLSs, how they work. That's nice. It's not just an exercise in informatics. It follows the biology, which it has to, or else you're wrong. You're doing the wrong coding. So I got excited about that one. It's a brand new one. Check it out if you're interested. All these other tools are in here, but again, these are tools for the PI, for the biologist to use. Okay? So you don't have to just toss your data over the wall all the time and then wait. You can just sit in your own living room, have a glass of wine or sake or whatever you're drinking, and do some analysis. So that was a long-winded example of a roadmap. Thanks for your attention.